Joseph, ECR, working on disease and pest detection. In this three-part video, I will be presenting the importance of seaweeds to the Philippine aquaculture industry, the common health problems observed in the farm, and the factors contributing to the occurrence of disease and pests on farm seaweeds. Seaweeds are the leading aquatic commodity in the Philippines, and the country is one of the top seaweed producers in the world. In 2019, seaweeds comprise more than 60% of the total aquaculture production volume in the country. That is 1.49 million metric tons out of 2.36 million tons of aquaculture production. Also, seaweeds and seaweed byproducts are the second highest contributor in terms of export receipts, amounting to $250 million after tuna. Seaweeds produced in the country are mainly from Caphaphycus alvarezii and Caphaphycus striatus, commercially known as cotonine, and Eucheuma denticulatum, commercially known as spinosum. These eucheumatoid red seaweeds are processed to obtain a hydrocolloid extract called carrageenan, valued for its many applications, mainly in the food industry, with their gelling, emulsifying, and stabilizing properties. Also, carrageenan is being used in other industries with pharmaceutical and nutraceutical applications. Other seaweed species, including green alga caulerpa, is produced mainly as direct human food. The increasing demand for seaweeds and their byproducts resulted in expansion of farming areas benefiting thousands of families, especially in the rural coastal communities. Seaweed farms are primarily small-scale and family-owned. Many of these farms are located in shallow water areas employing the fixed-off bottom technique, while others are in deep water areas including longline, multiple longline, and hanging longline methods. However, intensification of farming, limited sources of quality seedlings, and lack of biosecurity measures have affected seaweed production. After the country registered the highest production in 2011, a noted decline in production was observed. Several factors contributed to the decrease in production, such as seaweed health problems. Outbreaks of disease and pests, including ice disease and epiphytic pests, have significantly impacted the seaweed biomass yields and carrageenan quality, thus resulting in production losses and a decrease in income for the farmers.
Hello, I'm Jonalyn Mateo from Work Package 2 Farm Management and Legislation.
In this video, I will bring you to the world of seaweed farming.
First, I'll introduce you to the history of seaweed farming in the Philippines. Dr. Dote from University of Hawaii, who has been very active in the seaweed world, specifically with carrageenophytes. He had Filipino graduate students to become pioneers in seaweed farming, namely Dr. Gavino C. Trono Jr. of UPMSI, who was later known as the father of Caphaphycus farming, and Mr. Vicente B. Alvarez, who was the source of the species name alvarezii. They have tried to culture seaweed in different parts of the country. First, in Ilian Island, Mindoro Occidental. Second, in Panagatan Island, Caluya, Antique. Third, in Kiniluban Island, Palawan, until they have decided to move to the southern part of the Philippines and started planting in Sacol Island, Zamboanga, Tapan Island, and Sitangkay, Tawi-Tawi. Production has then started to increase. As we all know, the Philippines is an archipelagic country, and the characteristics of the coast are very different to each other. This is a seaweed farming map of the Philippines with GlobalSeaweedSTAR sampling points. Different farming techniques have been developed under two classifications: deep sea and shallow farming techniques. Deep sea farming technique can only be practiced in greater than 4-meter area during lowest tide. In the Philippines, we have different types. free swing or palabad, triangular, single vertical floating or tumbo-tumbo, hanging long line or spring, spider web or ambian, multiple raft long line or alul. A simple deep sea technique should have these materials: sinker, main line, culture line, floater. Shallow farming can be practiced below 0.3 meter during lowest tide. Fix off bottom or parasdas, single bamboo raft or palutaw, inverted umbrella or untang-untang, and these are the materials: stakes, floater, culture line, and a bamboo for a raft. There are also ecological requirements in seaweed farming. Site selection criteria: sandy, muddy substrate, protected from turbulent waves, moderate current, depth of water during lowest tide 30 cm, distance from freshwater runoffs around 500 meters, free from domestic pollution and agricultural runoffs. Environmental parameters: light intensity, water motion, salinity, water temperature, nutrient. By now, you already have an idea of how eukumatoids are farmed here in the Philippines. Enjoy farming!
Hi everyone! I'm Rema Sibonga, working on the characterization, conservation, and exploitation of algal genetic resources towards crop improvement. In this video, I will show you what we do during fieldwork. Our team went to different sites in the Philippines to collect seaweeds. We collected wild and farmed seaweeds, specifically the eukumatoid species or the Capophycus and Eucuma species. The sites where we collected samples were in Nampulugan Island in Sibuna, Guimaras, and different islands in Bohol, specifically in Danahon, Guindacpan, Mahanay, and Hao. We also collected samples from Samal Island in Davao del Norte. In Zamboanga, we collected from Layag-Layag, Tigtabon, and Arena Blanco. We also collected samples from different sites in Palawan, specifically in Puerto Princesa, Balabac, Quezon, and Taytay. In Iloilo, we collected samples from Ginasyan Ahoy. We also collected seaweeds from Tundon and Simonol in Tawi-Tawi. We have collected a total of 97 wild and 415 farmed seaweeds. In the field, we noted the GPS coordinates and other environmental parameters such as water temperature, salinity, pH, dissolved oxygen, and light intensity. After collection, we individually describe each sample. Each sample has its own identity. We assigned a specific code for each seaweed sample collected. We noted the description of the whole plant such as color, mode of branching, thallus shape and texture, and apical tip shape. We also recorded the diameter of the holdfast, the main thallus, and the different branches. From each sample, we cut a small piece of the apical tip which will be used for DNA extraction. The remaining plant were either pressed in herbaria sheet or air-dried. The herbaria and air-dried collection were placed at the University of the Philippines Visayas Museum of Natural Sciences. The GlobalSeaweedSTAR seaweed collection is readily available for other researchers and students who wish to conduct studies related to seaweeds.
I am G. Grace Suyo and I am an early career researcher for Work Package 4, which is the component of the GlobalSeaweedSTAR program that is responsible for analyzing the socioeconomic resilience of the seaweed industry. In this video, you will meet the lead characters in an animated story that we have created for you. They are Juan and Juana. and together they will share with you their activities and experiences in seaweed farming.